Hello and welcome to our webinar. This is Jeff DePinto from BangsLaboratories. I'm the Director of Sales and Business Development for Bangs Labs, and I'll be the moderator for this talk. Our topic today is Don't Worry, Bead Happy, Essential Knowledge for Microparticle Use in DNA and RNA Analysis. We have two speakers today. Our first speaker is Dr. Mitch Gore. He's a field applications manager at IDT. Mitch earned his Bachelor's of Science degree and Master's of Science in Animal Science at Virginia Tech and his PhD in Animal and Dairy Sciences from Auburn University. He then held postdoctoral positions at the University of Alabama College of Medicine in cardiology and at the University of Wisconsin in kinesiology. Mitch was previously the Director of Molecular Biology at Cruachem Incorporated and then Director of Life Sciences Technologies and New Business Development at PolySciences Incorporated, which is the sister company of Bangs Labs. In these roles, he concentrated on the development and commercialization of new tools for cellular and molecular biology research. Currently, as the Field Applications Manager for the custom oligonucleotides, gene expression, and genotyping portfolios at IDT, Mitch leads a global team that supports customer application needs in academia and corporate research, as well as for molecular diagnostics. Our second speaker, Kathy Kilbride, is the Director of Technical Service and Marketing at Bangs Labs. Kathy possesses a broad background in life sciences and quality assurance and has extensive experience working with clients developing novel and traditional particle-based diagnostic assays and bioseparation processes. Her expertise is in the optimization of chemical modification, ligand coding, and fluorescent labeling of magnetic and non-magnetic microspheres. Kathy's authored a number of papers and presented at scientific conferences, and she and her team routinely assist investigators with their research and product development efforts utilizing microparticle technology. Also, following the webinar, please add any questions that you might have to the chat. Any questions that aren't answered during the live Q&A session will be followed up later via email. And with that, I will pass it over to Mitch.
Thanks, Jeff, and thank you all for attending the webinar today. For my part of the webinar, I'll have a brief introduction to IDT and then go over some of the basics as they relate to using oligonucleotides with microparticles. So we'll touch on the topics of nucleic acid isolation, hybridization basics, oligo attachment chemistries, and then go over some scenarios that are related to genotyping, isolation of nucleic acid binding proteins, APTAMERS, and IMMUNO-PCR. So by way of introduction, IDT was started by Joe Walder in 1997, and as you can see from this timeline, IDT has evolved from simply a supplier of custom oligonucleotides to a solutions provider in the fields of qPCR, NGS, synthetic biology, and functional genomics. IDT has manufacturing locations in Coralville, Iowa, San Diego, Research Triangle Park, Leuven, Belgium, and Singapore, as well as numerous sales offices. We like to say that the sun never shines on IDT. In 2018, IDT became an operating company of Danaher Corporation and joined other life sciences companies owned by Danaher, such as Paul, Cepheid, Leica, Beckman Coulter Life Sciences, Cytiva, Molecular Devices, Phenomenex, and SciEx. So the core expertise at IDT is indeed oligonucleotide synthesis. However, from our customers' point of view, the need is to provide reagents that help them conduct their research at the highest levels of time and cost efficiency. Now, to this end, we've developed oligonucleotide products tailored to those applications and have developed bioinformatic tools specific to the design of oligos as they are used by our customers. So even though IDT is continuing to provide products formatted for specific applications, central to this is really the ability of IDT to control the quality of the oligos made at the IDT facilities. Now this is depicted in the graph where we're plotting full-length product percentages of plain desalted, not purified oligos versus oligo length for 3 different coupling efficiencies. The higher the coupling efficiency, the higher the full-length content. IDT controls the synthesis process such that a high coupling efficiency is obtained on the main IDT oligosynthesis platforms, and our highest coupling efficiency platform is used to produce IDT ultramers, the longer length oligos that are needed for many of today's applications. So now we'll move into the specific applications we wanted to discuss today, the first being nucleic acid isolation. So the goal in most nucleic acid isolation processes is to achieve as high a purity as possible, keep costs low, and to complete the process as quickly as possible. As for the use of microparticles with these processes, nucleic acid isolation is often done with magnetically responsive particles, allowing for the use of a mobile solid phase that can be manipulated via magnetic fields. This mobile solid phase lends itself to automation and is generally more efficient than static solid phase methods. Now, with nucleic acid isolation, there are two main methods: the boom method, named after the originator of the method, and SPRI, which stands for solid phase reversible immobilization. In the boom method, we are binding nucleic acid to a silica surface in the presence of chaotropic salts, and for SPRI, we're using carboxyl-coated microparticles to isolate the nucleic acids. Now, in the boom method, the magnetically responsive microparticles would typically contain a superparamagnetic precipitate made via the combining of two iron salts to create an Fe3O4 complex. Since it's superparamagnetic, that means it's responsive to a magnetic field, but it does not itself become magnetized. The Fe3O4 microparticle is then coated with silica to create a mobile solid phase that is used in the nucleic acid isolation process. So nucleic acid will non-specifically and non-covalently bind to the silica in the presence of a chaotropic salt, and after washing away all the unwanted material, be eluted in low ionic strengths buffers or water, ready for the next application. With SPRI, nucleic acid non-covalently binds to microparticles containing carboxyl surface groups in the presence of polyethylene glycol and salt. Now, like with the boom method, nucleic acid is then eluted under low ionic strength conditions, so it's ready to go in any application downstream. In SPRI, though, changing the ratio of the SPRI bead solution relative to sample volume will allow for site selection of the bound and subsequently recovered nucleic acid in the sample. Once we have isolated the nucleic acid from the tissue of interest, we then need to move on to analyzing the sample. This usually involves hybridization of one nucleic acid sequence to a sequence in the region of interest. This section will cover the basics of hybridization thermodynamics. Now, as mentioned before, sequence-specific hybridization is central to many molecular biology assays. In the example depicted here, oligonucleotides complementary to a target region are synthesized with a 5' biotin and allowed to hybridize to genomic DNA as part of an NGS library preparation protocol. So once the oligos have hybridized to their target, they are then isolated from the rest of the genomic DNA via the use of magnetically responsive streptavidin-coated microparticles. This type of hybridization capture is more efficient in solution versus static solid phase and is extensively used in many different applications. Now, crucial to these processes is the understanding of the factors influencing hybridization, such as melting temperature and secondary structure formation. Melting temperature, or TM, is defined as the temperature at which an oligonucleotide is 50% annealed. DNA concentration, salt concentration, and the sequence itself will all influence TM. So now let's look at those factors in more detail. So the graph in this slide shows several lines that are used to show the relationship between sodium ion concentration and TM. The red, blue, and green linear lines are from prediction equations based on experiments that predated PCR and are using data actually from E. coli genomic DNA and much higher salt concentrations than we use in PCR and other applications today. In some cases, these TM prediction formulas are actually still in use. The black curvilinear line is derived from experiments conducted at IDT using oligonucleotides. Of course, they are much shorter than genomic DNA, the original studies, And they were done using salt levels that are relevant to PCR and other molecular biology applications. And as you can see, these data show that the actual TMs are generally higher than those predicted by the other equations, and that TM relative to sodium concentration is not a linear relationship. So here we are examining the influence of monovalent and divalent salts on oligonucleotide TM. The graph gets pretty bumpy, depending on where you are on the axis showing, which shows the complex influences of salts on hybridization, making prediction of TM values difficult. It helps us a little bit in that most of the reactions that we do in molecular biology today occur over pretty narrow ranges of monovalent and divalent salts, as you can see by the bracketed areas there on the bottom axes. Here are some more factors that influence TM prediction. So on the left side, we are looking at the influence of oligonucleotide length and GC content. The longer the sequence, the higher the TM. Also, as GC content increases, we also increase TM. In this example, you can see easily how a 30-degree temperature difference can exist between two oligos. On the right side, we are looking at the influence of DNTP concentration on TM, where DNTPs depress the TM of oligos in hybridization reactions. Based on what we have shown in the last few slides, it's evident that TM prediction is very complex, thus the use of algorithms that allow the prediction to be made in consideration of all these factors is important. So to help with TM and other predictions, IDT has a suite of tools you can use. These are available on the IDT website at no cost to the user. To access them, or to access SCITOOLS, click on Tools near the top of the IDT homepage. So once you click on Tools, you'll have a screen with several options. Now, for the purposes of the discussion today, we'll focus on OLIGOANALYZER. Now here we have the OLIGOANALYZER tool interface. We'll use it to show how influential magnesium and DNTPs can be in predicting TM. So if we type or paste the sequence in the box at the top, and then leave the values for magnesium and DNTPs at the 0 level, which is the default level on the program, you can see that we get a TM prediction of 58.5 degrees. So on this slide, we've added in the magnesium and DNTP levels that are relevant to hybridization reactions, and in this case, it's PCR. So now we have a TM prediction of 64.9 versus the 58.5 that we had previously. This large difference in TM is enough to affect the specificity and therefore the accuracy of the hybridization. With the OLIGOANALYZER, not only do we get a feel for the TM of the sequences we are working with, but often we also need to know how likely they are to form secondary structures that lower actually the amount of sequence that can participate in the hybridization reaction. Here we're looking at the likelihood of a sequence to form a hairpin structure. As with TM prediction, the hairpin formation is also influenced by the ionic environment of the oligonucleotide. In this case, we can see that the sequence could form a hairpin and that the particular structure are associated with Gibbs free energy values in the tool. In general, the more negative the delta G value, the more likely that structure will exist in solution. So the negative Gibbs free energy value here isn't terrible, but as a rule of thumb, anything that has a delta G of -9 and below is worth looking into and avoiding if at all possible. Now, another aspect of working with oligonucleotides and microparticles is covalently attaching them to the microparticles themselves. This section will go over some of the main considerations when considering how to do these types of reactions. So one can use a variety of surface chemistries. Listed here are the main ones available for use with oligonucleotides: carboxyl-amine, chloromethylamide, hydrazide, aldehyde, hydroxyl, thiol, and epoxy groups can all be utilized, but in practice, the primary attachment chemistry strategies utilize primary amine groups. Here we have the common amine linkers available on oligonucleotides. They differ by the length of the carbon chain and their placement in the oligonucleotide sequence. C6 and C12 linkers are available, as well as 5', 3', and internal linkers. When using primary amines, you can use several different types of attachment chemistries, such as attaching primary amines to isothiocyanate groups, succinylmethyl esters, or sulfonyl chlorides. While useful, these chemistries are really not often used with microparticles. The most common attachment chemistry when coupling oligonucleotides to microparticles is carboxyl-to-amine coupling. Typically, the microparticles are made of polystyrene polymers created in an emulsion polymerization reaction using acid monomers, which give the microparticles carboxylic acid surface groups. These carboxyl groups are reacted with the primary amine groups on the oligonucleotide via a carbodiamide-mediated reaction. The carboxyl group reacts with the CN bond of the carbodiamide to form an O-acetylated urea derivative, which then reacts with the primary amine on the oligo to form a stable amide carbonyl group. So now we'll switch gears and go over some of the basic approaches for microparticle-based molecular detection of mutations in nucleic acid sequences or genotyping. So when we are detecting single nucleotide polymorphisms or SNPs, we can break down the process to 3 parts: isolate and then sometimes amplify the target sequence that contains the mutation, perform a reaction that discriminates one allele from another, and then detect which allele is present. So when using microparticles in SNP detection, one of the most direct ways is via single nucleotide extension reaction. For this approach, an oligonucleotide that contains a complementary sequence to the target immediately upstream of the SNP site and a tag sequence that is not complementary to the target sequence. Also used is a labeled dNTP that is complementary to the mutation, and an oligonucleotide conjugated to the fluorescent microparticle that is complementary to the tag sequence on the allelic discrimination primer. If the mutation is present, a polymerase adds the labeled dNTP to the allelic discrimination primer. This now labeled oligonucleotide is captured by the oligo attached to the microparticle. This whole complex can then be detected via flow cytometry or other methods. So this slide is an extension of the previous slide where multiple labeled nucleotides are incorporated as the polymerase copies the target sequence, and this increases the amount of signal available for detection. This process can also incorporate the use of microparticles containing 2 different fluorescent dyes, and oligos conjugated to those microparticles are complementary to 2 different tag sequences. This allows for the simultaneous detection of 2 or more alleles in the same sample. Now you can also do SNP detection using ligation-dependent mechanisms. So in this approach, 2 oligonucleotides are used that have perfect matching around the SNP site. So, and one of these oligos will have a tag sequence. Now, if they match perfectly, then a ligation reaction can occur using DNA ligase, resulting in a template sequence that can then be amplified and fluorescently labeled by performing a PCR reaction. The amplicon from the PCR reaction can then be captured by an oligo conjugated to a fluorescent microparticle, which in turn can be detected by flow cytometry. Studying nucleic acid binding proteins such as transcription factors can also be done via the use of oligonucleotides and microparticles. This section will cover 2 of those approaches. So for DNA-protein interactions, one of the assays is the avidin-biotin complex DNA binding assay, or ABCD assay. So in the ABCD process, the biotin of the oligonucleotides are annealed to form the target sequence for the DNA binding protein. This double-stranded DNA and the protein extract are then allowed to interact. After that incubation, where the protein binds to the double-stranded DNA, magnetically responsive microparticles that have been conjugated with streptavidin are added, and the DNA protein complexes are collected for downstream analysis. Chromatin immunoprecipitation protocols take the approach of using biotinylated antibodies to DNA binding proteins. Here, DNA protein complexes are allowed to interact with biotinylated antibodies to the proteins of interest. The attached biotinylated antibodies are then allowed to interact with magnetic particles conjugated to streptavidin for magnetic collection of the DNA protein complexes. So to wrap up the IDT portion of the presentation, I wanted to mention 2 fairly recent developments that utilize microparticles, APTAMERS and IMMUNO-PCR. Aptamers are oligonucleotide sequences that can form secondary structures that mimic ligands that bind to receptors in other molecules. In this example, an aptamer and a magnetic microparticles are used to detect ochratoxin A, a food toxin that can be a contaminant in grains. In this assay, binding of ochratoxin A causes dissociation of the aptamer from a poly-T, poly-A complex oligo that's been conjugated to magnetically responsive microparticles. The dissociated aptamer is then isolated from the poly-T, poly-A microparticles and quantified by qPCR. IMMUNO-PCR is an ultrasensitive method to detect proteins. The process can be further enhanced using magnetically responsive microparticles. So in one version of this approach, magnetic microparticles are conjugated to a detection antibody that's directed at the protein of interest, and then that is allowed to interact with the sample. In the next step, a detection antibody, which has either been conjugated to a PCR capture oligonucleotide sequence or is on a particle containing capture sequence, is allowed to bind. The bound complex is enriched via magnetic capture and detected via the detection oligo hybridization and subsequent PCR. The very large ratio of captured DNA sequences to antibody and the amplification of the signal by PCR results in an enhanced sensitivity for an already ultrasensitive method. So that concludes the IDT section of the presentation, so please feel free to contact us if you would like additional information. Also, thank you very much for your time and attention. So now I will pass it over to Kathy for the BangsLaboratories portion of the webinar.
Thank you, Mitch, and hi everyone. Thanks for staying with us. So I'm Kathy Kilbride with Bangs Labs. I serve dual roles in tech support and marketing, and I'll be picking up the second portion of our Don't Worry Bead Happy webinar. And basically, I'll be speaking to all things beads, from selecting them to coding and QC, and showing how these can aid in informing this part of your development and manufacturing efforts. Now, before we dig in, just a bit of a further introduction. Bangs Laboratories is part of the Scientific and Polysciences family of companies, and you can see us there as the leftmost pin on the map. The other markers show locations of Polysciences sales and manufacturing facilities in the U.S., Europe, and Asia. And this is an image of a picture of our facility near Indianapolis. This houses our administrative, client support, laboratory, clean room, and manufacturing spaces. And more importantly, what do we do? Well, we manufacture all different sorts of microspheres. You can see in the images, the gray images from right to left, silica, polymer, and magnetic microspheres. We also can dye polymer beads internally with visible dyes and fluorophores. And we also coat microparticles with all sorts of ligands such as antibodies, antigens, peptides, and of course the nucleic acid probes. And it's these sorts of modifications that ultimately make the particles useful, especially in the diagnostic space. Our beads are used in a broad range of applications. You can see here a variety of diagnostic tests and assays, different sorts of bioseparations, including things like nucleic acid isolations or cell or antibody separations. And additionally, microparticles are used as analytical instrument standards. And in particular, we manufacture a lot of products for fluorescence-based technologies such as flow cytometry. This is a short overview. The topics I'll be covering include particle characteristics and characterization, selecting the right particle for your application, and of course, the really important one, making it work. So considering the first part, which is particle characterization and particle characteristics and characterization methods, we're really talking about the raw materials that you'll be obtaining from a supplier. it's important to really understand what you have to work with and also how to assess these raw materials. If there are parameters, for example, that are really important to your process or assay but have not been characterized. So we're going to look at material and composition characteristics, some general characterization methods, and then also application-centric characterization. Finally, it'll be a little bit later, but I'll talk about some methods for your finished reagent evaluations that you'll be conducting in-house. But really the point of this, just to punctuate it, is that going into your method or reagent process, you really need to know that the particles have the needed characteristics to support your requirements for the application in general, of course for your reagent coding or manufacturing processes, and also specific assay or application performance requirements. Now, considering material and composition characteristics, every type of particle has different physical, optical, and mechanical properties that will be inherent to the composition and potentially the synthesis method. In the example here to the right, I have images of silica, polymer, and a magnetic particle. These are just demonstrating high sphericity, the smooth surface of these particles, or a rougher surface here. These are all things that are going to go into your surface morphology and your surface area that's available for binding reactions. In this example, you can see that different materials have different binding capacities. Silica binds the lowest amount of a representative protein. In this case, it's streptavidin. Polymer spheres bind kind of a mid-range amount of protein. And then this magnetic particle, which is a polymer-based magnetic particle, binds high amounts of streptavidin. And this is evidenced by just single particle binding by 250. And so just on the basis of the material itself, of course the size and the surface characteristics, we're going to be able to kind of have an idea in advance of what the potential binding capacity is. Other sorts of things that the material will dictate will be hydrophilicity or hydrophobicity, charge, density, solubility parameters, refractive indices, absorbance characteristics, and again, surface morphology or roughness. Considering some general characterization methods, these will be things that the manufacturer can do, can really tailor to their own manufacturing specifications or to your specifications. These will be things like surface functionality, the diameter and size distribution, and you'll be able to obtain lot-specific data from the manufacturer. In this example, you can see the top particle is highly spherical. It's very uniform in size, and here's a sizing histogram to the right. In the bottom example, it's a different magnetic particle, about 1.5 microns in size, but this sizing histogram has a log scale, so you can see it's actually quite a polydispersed size profile for this particle. So again, just from this very general characterization, you can find that particles are well intended or not for certain applications. Other types of characterizations that will be done will be very application-centric. These may be certain sorts of analyses that are done by the manufacturer, performed by the manufacturer, or these may be things that are not characterized by the manufacturer, but that you'll need to assess in-house. These can be things like intrinsic background of the material and how that would impact your assay. So that could be something like chemiluminescence, free iron, autofluorescence, or fluorescence carryover, magnetic separation profile if it's a magnetic particle. How much time does it take to separate? How uniformly do the particles separate? Distribution of surface groups and coating homogeneity. We also sometimes look at things like light scatter properties and, of course, special properties like fluorescence or visible color. In this example to the right, this is flow cytometry printout, has a dot plot and fluorescence histograms below. This is from a run of a 6-micron polymer magnetic bead, and this is really just an autofluorescence check for downstream fluorescence applications. If you look at the histograms below on the x-axis, moving to the right, that would be increasing fluorescence intensity. So because all of these histograms show the population is seated near the leftmost y-axis, or it hasn't progressed along the x-axis, this is an indication to us that this particular bead has very low autofluorescence and would be well suited for flow cytometry applications or other sorts of applications requiring low auto signal. So those were just some basic sorts of material and particle characteristics and characterization methods that can be used to screen beads. And so now that you know something of these different materials and characterization methods, I'm going to speak a little bit about how to specifically apply these to selecting the right candidate for your application. And by right particle, we really mean the best candidate for testing and screening. And I'll go over a few different applications, particle selection, 4 different applications. So those include SPRI, nucleic acid isolation, chemiluminescence assays, and also time-resolved fluorescence lateral flow assays. So in this first example of bead selection for SPRI-based nucleic acid isolation, as Mitch described, the process involves using PEG and salt to condense DNA onto a magnetic particle, a carboxylated magnetic particle. That after a binding event, the particles are isolated, a wash occurs, DNA is eluted, and then it can be utilized for downstream applications. The bead characteristics that are going to be important for successful nucleic acid isolation by SPRI include a carboxylated surface, because that surface participates in an ionic interaction with the condensed DNA and PEG complex. Surface area is, of course, going to be important. This is going to be the diameter and size distribution of the particle population. Also, potentially bead topography, because really what we're looking for is the best scenario for high and efficient binding of that DNA-PEG complex, and also good elution characteristics. Another characteristic that's going to be really important is magnetic separation rate and uniformity of the separation, because a lot of these processes now are automated. There's an automated nucleic acid isolation workflow. And so just considering one magnetic particle, our Magnify carboxylated magnetic particle, some of the characteristics that make this particle suitable for the application, of course, include its carboxylated surface, and we do characterize that through surface titrations. Particles are, of course, sized. This is about a 1 micron particle. And you can also, if you view the SEM image of a representative bead, you can see that the surface topography is such that there is a certain amount of surface roughness which lends a greater capture area for the particle. And also, there aren't such deep crevices on the surface of this particle that elution becomes a problem where DNA might get trapped in those crevices. We also do a basic DNA isolation with the particle by a SPRI just to ensure that the characteristics are performing as we expect. And we also measure magnetic separation rates of each lot that is produced. So again, this is going to be an instance where we do a lot of characterization at our facility because we do anticipate clients placing this particle in this specific application. And if you want to view more of this data, we do have a poster available on our website. So in the second example of particle selection for a specific application, I've chosen a chemiluminescence assay. It's much like the assay that Mitch selected. It's an toxin A assay, but in this case, it does have a chemiluminescence detection. In this example, probes are conjugated to a magnetic particle. An aptamer is then synthesized that is specific for ochratoxin A. This aptamer will complex with the probe on the bead in the presence of phenyl glyoxyl. Then a chemiluminescent signal is generated because that reacts with guanidine bases in the aptamer. In the competitive format, in the presence of ochratoxin A, the toxin binds to the aptamer. The aptamer that doesn't bind to the probe on the surface of the bead, so there is a lesser chemiluminescent signal generated. So the bead characteristics that we're going to be interested in for this sort of an assay will include, of course, basic surface functionality. Also, surface area will be important. This is going to be the diameter and distribution of the particle population, also considering what the topography of the bead may look like, because both of these together result in the binding of an appropriate amount of the probe that we want to immobilize. We're going to be interested in whether or not there's basically intrinsic background in the particle that could interfere with the detection of the chemiluminescent signal. And then also, a lot of these assays are going to be automated. So the magnetic separation profile, again, the separation rate and the uniformity of the separation will be important to support the automated assay workflow. In this example, we have a ProMag HP magnetic particle. Of course, we do appropriate sizing, and you can see the surface characteristics of this particle. It does have some surface roughness to it, but not an extensive amount. So we do get increased surface area, but again, not such deep crevices that we might be losing probe or running into steric effects because probe is interacting with those crevices too much. We also assay basic background of these particles. We're not looking at exactly the same system here, but we do screen them for exposed iron because iron is a known interferent with chemiluminescent signal. And also we do a basic chemiluminescent screen with the particles. Finally, we do basic magnetic separation rates just to ensure that each lot of beads is going to separate quickly and completely to support that automated workflow. Now, in particular for chemiluminescence assays, there really isn't a one-size-fits-all magnetic particle or particle that is going to fit every single assay. There are certainly different application areas, different recorders are utilized, so background should be tested in different ways. Certainly there are many different chemiluminescence platforms. And so in this instance in particular, it is really important to both identify good candidates for screening, but probably screen a number of particles to ensure that you're getting the optimal performance characteristics from your reagent. In the final example of particle selection for a specific assay format, I'm going to address particles for time resolved fluorescence lateral flows. This is going to be the traditional lateral flow format where the particle wicks through the membrane with the addition of sample and then is captured at both a test line and a control line. But detection is going to be conducted typically with a handheld fluorescence reader that is able to measure time resolved fluorescence or fluorescence over a specific amount of time. There are many quantitative assays that are being developed utilizing this format, and specifically for things like influenza and RSV. So when selecting particles for this type of an assay, of course we need basic surface functionality. We're going to be interested in both specific and non-specific binding because the particles not only need to bind our ligand and target of interest, but they're interacting with physical components of the device, such as the membrane. We're going to be interested in the diameter of the particle and its size distribution, so that we have appropriate surface area and it wicks through the membrane appropriately. And also, in this case, beads need to be dyed with a reporter that exhibits a defined time-resolved fluorescence signal. And this is going to feed into our sensitivity and dynamic range of the assay, but also we need to be assured that the particle fluorescence signal exhibits suitable stability. And just as a couple of points of characterization, you can see that here's a sizing histogram that shows a highly uniform diameter of the particles. And additionally, you want to be assured that particles have appropriate fluorescence signal from lot to lot. So in the third and final segment of this, we're going to come back to characterization strategies that will help you to make it work. We're going to be talking about bead coating strategies, as Mitch did a bit, and also particle reagent evaluation. So this is going to be evaluation that you would do in-house of your finished reagent. Mitch described a couple of bead coating strategies, and again, these are typically going to be covalent immobilization of an amine-modified oligonucleotide. So basically, you'd have a carboxylated bead that would be activated with a carbodiamide like EDAC. You would add the aminated oligo, bind it, do a wash, and have covalently bound probe to your particles. Another common method is the streptavidin biotin method, where to your streptavidin coated beads, you would add a biotin modified oligonucleotide. And both of these will result in essentially a permanent bond to the particle. Because it's an oligonucleotide and has a specific attachment point, the attachment specificity is going to be very good. It is going to be immobilized at a single end of the oligo. And while there are points of optimization with more with a covalent reaction, there's really not a lot of difficult optimization required. So that's not to say that there aren't opportunities for optimization and customization, and that would be also in consideration of your specific assay or application. Some considerations going in will be the length of the probe, the coating density, and how that impacts capture or hybridization efficiency. Looking at the example immediately to the right, here's an illustration of probe length versus packing configuration. And you can see that in this study, investigators found that long probes tend to coil on the surface in sort of a polymeric configuration, whereas shorter probes extended out into solution in a more linear fashion. And so sometimes to combat this, one thing that has been done is to, if you take a look at the graphic below, actually immobilize shorter chains between the longer probe. This can be done both for nonspecific binding purposes or to really get that, that probe extended out into solution so that it can hybridize efficiently. So in some cases, this might be a different sequence that's just nonspecific and it's a shorter sequence. In other cases, it could be something like a peg chain. This also, I think as I mentioned, can have a clocking function. So after you do your bead coatings, you've developed your reagent, it's going to be important that you evaluate them in-house immediately following the coating, of course, to assess the quality of that, but on an ongoing basis to assess stability of the coating. Some other kind of criteria that you'll want to consider will be the dispersity of the suspension. In this first example to the right, you can see just using a light microscope, you can often determine if the suspension is monodispersed or if there's something stickiness or aggregation. In the second example to the right, another thing you'll want to look for can be contamination over time, and this is really a gross contamination that I'm showing you, but your microscope can also identify that. Other sorts of things that you want to assess will be the coating activity and the binding capacity, and also uniformity of the coating. So looking at the lower two images, the fluorescence histogram from a flow cytometer, the top histogram demonstrates a coating that does not have good bead to bead consistency. So this would be important for single bead assays like a flow cytometric assay or other, maybe a microfluidic based assay where you really are looking at every single particle. This tail indicates that the binding is very heterogeneous in some population of the particles. The lower image does not have that tail, and so it shows that from particle to particle, we really are getting a very consistent binding response. And again, you'll want to look at the particles over time to ensure that you have shelf life for yourself and for your customers. So this example demonstrates coating stability that was assessed over a 12-month period on a flow cytometer. So for 4 different beads in a particular kit, the signal was evaluated over a year's time. So ultimately, for bead reagent evaluation or even incoming materials, there are a lot of instruments and strategies that you can utilize for bead QC. A lot of these are going to be common things that you have in the laboratory. Again, like a microscope will tell you a lot about the general appearance of the beads and the quality of the suspension. If you're fortunate to have a flow cytometer, there are many things you can discern about the beads, from autofluorescence signal to dispersity to their binding profiles. And of course, there are other sorts of particle sizers. You can assess magnetic separation, specialized techniques as you would have a luminometer for looking at background chemiluminescence. Of course, surface titration, iron assays for background, again, in a chemiluminescence assay. And you can develop functional assays that will help you assess your coating characteristics, the development of your coating methodologies, and then actual performance in your final assay. So in summary, there are many benefits and limitations of different compositions. There are many different characterization methods, whether this is going to be something that is performed by the manufacturer or in-house. And these together will help you identify particles that are going to be the best for screening purposes and starting your development efforts, so that you're not starting down really the wrong path with the wrong particle. And then finally, when you do select particles for development and screening, there are many different things that you can do to modify your coatings if necessary, with things like tethers, blockers, what have you, to really fine-tune and optimize particle performance. So we do have a lot of additional resources on our website. Today was really just touching on a few topics that are important for particle collection and use. We have another webinar that you can view on microsphere coating. A few pieces on our website that may be of particular interest are some covalent coupling tech notes, and also some data sheets with protocols for strict iodine biotin binding. And of course, you can contact us for tech support anytime at info@BangsLabs.com. So thank you very much for staying with us, and we look forward to hearing from you.
Well, thank you, Mitch and Kathy, for some very interesting content, and thank you all for attending our webinar. We've received several questions from the audience, so I'll start with those specifically addressed to Mitch at IDT first. So the first question, Mitch, is how important is oligonucleotide purity when conjugating to microparticles?
Thanks, Jeff. The purity is important, although you have to think about the process of conjugating microparticles or conjugating oligonucleotides to microparticles. Typically, we're working with a 5' amine group, and since oligonucleotides are made in the 3' to 5' direction, only the full-length oligos in the synthesis will actually have a 5' amine group. So, in effect, the conjugation reaction itself is an effective way to get purified oligos, if you will, attached to microparticles. So, of course, you need a high-quality synthesis, and you want a lot of full-length content. But keep in mind, if the oligo is not too long, you don't really need to pay for extra purification if your conjugation reaction is actually going to do that for you.
Okay, thank you, Mitch. Very interesting. We have another question for you. How do I know how much oligonucleotide to use in a conjugation reaction?
Yeah, that's a little more difficult. The amount of oligonucleotide that you're going to use in a conjugation reaction should be in excess of, of course, the other reactive groups that you have. You can talk to the Bangs Labs guys about that a little bit, in that being able to estimate the number of reactive groups or the number of moles of reactive groups on a microparticle and sort of match that, or at least know what that target is relative to the number of reactive groups or moles of reactive groups on the oligonucleotide is the best way to go about it. Because really what you're trying to do is to dial in a little bit of access, but you don't want so much that you get a lot of nonspecific binding to the particle.
Okay, excellent. We have another question. You mentioned the SCITOOLS OLIGOANALYZER module. I'm interested in designing oligonucleotides for my experiments, and what is the best way for me to start, and can you help?
Sure, yeah, we're always happy to help, and at IDT we do have a scientific applications group, as well as the group that I'm a part of, the field applications group, that can help you in designing your experiments, at least as far as the oligonucleotides are designed. In the SCITOOLS set of design tools that are available on the IDT website, there's actually a couple of different ways to get at designing oligonucleotides for experiments. Most of those are directed at PCR, but of course the same thermodynamic parameters that are important for PCR are pretty much the same ones that you would want to consider for any other types of reactions or hybridization-based reactions. So you can use those tools like PrimerQuest on the IDT SciTools suite to design those.
Okay, great. Thank you, Mitch. I'm going to move on to some questions for Kathy. Kathy, the first question is, how similar is SPRI-based RNA isolation compared to the process for DNA?
Basically, they're going to be fairly similar. Sample prep at the beginning will, of course, have some differences. The binding mechanism is going to be largely the same. The real difference often comes in the buffers. The DNA buffer is typically going to be TE and tween, whereas the RNA buffer is going to be citrate and tween. And the aim for this is to minimize the base hydrolysis of the RNA through the use of sodium citrate. RNA buffers also tend to have a lower pH. I've seen like range of about 6.4, whereas DNA buffers are going to be higher pH at about 8.0. And RNA buffers are additionally going to contain DTP to inactivate RNases.
Okay, great. Thank you, Kathy. We have another question, Kathy. How can I quantitate nucleic acid bound to beads?
There are a variety of methods that you can use, and we will select a different method, both based on ease of the method and where we are in our process. So if you want to get a quick answer to just, was your coupling effective from a yes-no standpoint, or kind of basically successful, you can use photometry and just do an A260 on what didn't bind to the beads from a post-binding reaction. From there, you could move on to a fluorescence-based reaction. And you can use, there are a lot of reagents that will bind either double-stranded or single-stranded DNA or RNA. You can look at what didn't bind in solution, or you can look at bead-bound nucleic acids using flow cytometry. And then, of course, you can develop a functional assay, which is going to be, really when you've been through the other stages and you're ready to test it in the final application.
Excellent. Thank you for that great information, Kathy. Unfortunately, that's all the time we have for questions. As a reminder, any remaining questions that weren't answered during the live Q&A will be followed up by email. I'd like to thank everybody once again for attending the webinar, and have a nice day. Thank you.